Hello, my dear students. So, welcome to our next lecture in PM1A. This lecture is going to be on blood grouping. This is a very interesting lecture because we are going to be talking about our own blood groups and why they are important and when they are essential and all the other facts about blood grouping. Okay, so the learning outcomes for this lecture. At the end of this lecture and after further reading, you should be able to describe in detail about ABO blood grouping. What is ABO blood grouping and why it's important. And then you should be able to understand the significance of rhesus blood grouping, because that's what is differentiating between negative and positive blood groups and why it's important, its significance for different pathological conditions or during blood transfusion. So you should be able to understand all these things. Then describe the methods used for blood grouping. What sort of methods we are using, how we are determining the blood grouping, and as a pharmacy student, why this is important for you guys to understand how blood grouping is done and the significance of blood grouping in clinical settings. Then you should be able to demonstrate the importance of blood grouping during blood transfusion, because that's when we must make sure that the blood grouping is matched and we are double checking, triple checking their blood groups between the donors and the recipients. Then in addition, you should be able to understand the importance of blood transfusion, compatibility and incompatibility. So prior to your transfusion, you need to check whether the donor and the recipient are compatible or incompatible, because if they are incompatible, you can't give the transfusion. It will cause a lot of problems. So that's what we will be talking about in this lecture. OK, as usual, I will try to explain everything in an easy manner. And if you don't understand anything, please feel free to send me an email and I'm happy to go through them, or we can discuss during our tutorial session. OK, there are different ways of blood transfusion, so you shouldn't think that whole blood is collected from donor and straight away the whole blood is going to go into the recipient. OK, that's not the case always. In some cases, people will infuse the whole blood, collected, tested, and then infused to the needy people. They are called recipients. In other cases, you can take different components of the blood. It doesn't have to be the whole blood. For example, we can take just the plasma. That's a liquid component of the blood, just the plasma, and then transfuse to some individuals who need only plasma, for example, clotting factors. So in these cases, in hospitals, mostly they will use fresh frozen plasma. And fresh frozen plasma can also be used to separate several important proteins from the blood, for example, coagulation factors, because some people are deficient for specific coagulation factors, and therefore it is really important to isolate some of those specific factors and infuse into them. And there are other uses for plasma as well. So we can use the whole fresh plasma or fresh frozen plasma or individual component of the plasma. And then you can take the cellular components, so mostly the red cells. For example, for people with anemia, we can infuse red cell concentrate, only red cells. And then for some people, only platelets will be infused. OK, so they will isolate the buffy coat. That's where all the leukocytes and the platelets will be upon centrifugation. And then from the buffy coat, you can separate out all the platelets and then infuse to people who need only platelets. So blood transfusion means it's not just the whole blood transfusion. OK, understand that. So in some cases, they will infuse the whole blood. In other cases, they will infuse individual components of the blood. So in the last lecture, we talked about the components of the blood in very much detail. So you can go through them and try to remember them for this lecture as well. OK, so what is the history about blood transfusion and blood donation? So the first successful blood transfusion was carried out in 1665 by Dr. Richard Lawyer, using mainly animals. OK, they collected blood from animals, infused into humans, and then watched what happens. These transfusions of blood in humans caused unexplained deaths. There were lots and lots of deaths happened because of these inappropriate transfusions. And then later, only in the late 18th and 19th century, Dr. Carl Landsteiner discovered the 4 main blood groups in humans. Okay, and he received a Nobel Prize for this discovery. So it's really important that Carl Landsteiner identified these blood groups, and after that there was so much of research gone into blood grouping, identifying different variants, alleles, and different matches, mismatches, all those things. But Dr. Carl Landsteiner was the first person to identify the main ABO blood groups that still we are using. Okay. So this is a schematic picture published in Lancet ages ago. So this is how in the past people used to transfuse blood straight away from the donor, straight into the recipient. No test, no storage, nothing in between carried out. And as you can imagine, this will cause lots and lots of problems. Okay, so that's why the blood transfusions and the blood donations are highly regulated these days, and strictly they follow specific measurements and specific storage conditions and specific various tests they carry out during donation and prior to transfusion as well, in order to avoid any inappropriate or unexpected consequences. Okay, so when we talk about blood transfusion, we need to talk about why do we care about blood donation and why do we care about blood transfusion. Okay, so these two terms we will be using interchangeably quite often, because blood donation is one side, the donors are giving the blood, and then blood transfusion is on the other side, recipients receiving the blood. So don't get confused between the donation and transfusion. So the blood transfusion is absolutely essential during the treatment of excessive blood loss, for example, due to injury or during major surgeries. Okay, if somebody is going for a major surgery in hospitals, the hospital will make sure that their blood group the matching blood is readily available, because during surgery people will lose significant amount of blood and therefore they do need transfusion. Okay, where else we need blood transfusion? During maternity. Okay, particularly during childbirth, there will be a lot of blood loss and these people need blood transfusion. During maternity, the blood will get diluted. Again, we talked about this in the previous lectures, and therefore during pregnancy, people may need some blood transfusion as well. And then during cancer treatment, particularly for chemotherapy treatment, because there won't be many blood cells after the chemotherapy and the production will be affected as well. Therefore, they need transfusions. And then patients with blood related disorders. Okay, there are several blood related disorders. Again, we touched on all these things in the previous lectures on blood clotting and introduction to blood. So there are some people who are deficient for specific clotting factors, some people who are deficient for platelets, and some people, for example, anemic patients, they have low counts of red blood cells. So all these people need blood transfusion. Okay, and as I said previously, it could be the whole blood transfusion as we collected from the donor, or it could be specific component of the blood. It could be cellular components, particularly red cells and the platelets, or it could be non-cellular components, for example, plasma or plasma proteins. So you need to remember all these things. So in the UK alone, around 7,000 units of blood or blood is needed every day. Okay, 7,000 units are needed. It's really important. Each unit is a point. Okay, that's around 568 milliliters. So we need quite a lot of blood every day to meet the demand during surgeries and for patients with other diseases and disorders who need blood transfusion. So annually, we need around 2 million units of blood or 2 million points of blood, and the blood donations, the NHS and the blood collection people or blood donation campaign people are collecting this blood from around 1.4 million donors every year. Okay, so in short, we need around 400 new donors every single day, or 135,000 blood donors, new blood donors every year, in order to replace the donors who cannot give blood anymore. Okay, so it's really important that we all play our role by donating blood, because there are so many people who need blood, and who knows, one day we may also need blood. when we get some diseases or during surgeries. So it's really important to register and donate blood to meet the demand. And at the moment, there is a high demand for Black ethnic people because the blood from these people mainly needed for transfusion on certain individuals. So there is a high demand. And as always, the RH negative blood donors are in high demand. Okay, we need RH negative bloods because it's a really rare blood group, which we are going to talk about later on. So therefore, we need more donors from RH negative blood group people. So these are the demand. And during blood transfusion, it's really important to ensure specific criteria for the selection of donor. Okay, so therefore people will be chosen very carefully to donate blood. So the hospitals cannot take blood from everyone who just want to donate because there's a lot of screening procedure and health surveillance. They have to go through all these factors to select the donor. So generally, people who want to give blood or the blood donors should be in good health. They shouldn't have many diseases or any notable diseases. It's really important that they have a good health. And it should be unpaid volunteers. Okay, if you're paying them, then they might come to give blood almost every day or every week, which is not acceptable. They need to wait certain time between the donations because it's quite a lot of blood, 568 ml. So therefore they need to wait several months before giving the blood again. And the hospital needs to exclude several risk factors. For example, the blood donor should not have a jaundice, hepatitis, or a travel during malaria, so travel in malaria areas, or other risk factors. For example, they shouldn't have any HIV and other things. It's really important. And then their blood has to be screened. And it's mainly for the blood groups and also to screen any of these notable diseases. So which we'll talk in the next slide. So it's really important to do the screening before taking the blood, otherwise their blood will not be useful and we have to waste it. So it's really important to screen all these things prior to taking the blood. And prior to the transfusion, the donated blood should be compatible with transfusion or recipient's blood. So the compatibility testing must be done prior to transfusion. So the hospital will do the compatibility test between the donor's blood that they collected and the recipient's blood who is going to receive the blood. So if it's matching or if it's compatible, then they will proceed for transfusion. So the blood we are donating will have limited lifespan, so it has to be used within that lifespan. Again, the hospitals or NHS will ensure that they are not wasting much of the blood donated from people. So for example, red cells, they need to be used within 35 days. Okay, so again, in the first lecture, we talked about the lifespan of these days. Red cells normally will be having around 120 days of lifespan, but the donated blood must be transfused within 35 days in order to avoid cell lysis or to avoid transfusing any low quality red cells. And the platelets, their lifespans around 7 days, and they had to be used within 7 days in order to benefit the recipients. And the plasma, fresh plasma mainly, it has to be used within 3 days. Sometimes they may store specific components isolated from plasma for some time in freezers, and it will be used in a later date. But mostly they will be used on time as quickly as possible. So these are the criteria and use date for the blood that we are donating. Okay, what are the routine tests that people do in the hospitals for donated blood? So the first thing, ABO blood grouping. So that's what we are going to be focusing on this lecture mainly. And then the RH blood group, that's called rhesus blood group, particularly RHT. That's what is determining whether we are positive or a negative blood. So these things will be done specifically. And then they will be identifying any antibodies produced specifically against red cells or any other notable diseases. For example, hepatitis B, it's really important, hepatitis C, HIV and treponema or syphilis or other diseases that are causing leukemia. And these days, obviously, COVID-19. So it's really important to screen for all these major risk factors and blood grouping prior to transfusion. And then finally, prior to transfusion, they will do the cross matching, as I mentioned before. So we need to ensure that the donated blood is matching for the recipient's blood. Otherwise, they will not proceed with the transfusion. They have to find another blood. So these are the routine tests that the hospital or NHS will do for the donated blood. Okay, so now we're going to talk about ABO antigens. So what are ABO antigens? Because these are the important antigens we will be talking about again and again whenever we are talking about blood donation or blood transfusion, because they are really important. Okay, so these antigens are present on the red blood cells. Don't forget that. Okay, so all the blood group antigens that we are going to discuss in this lecture are mainly present on the surface of red blood cells. Okay, so particularly RBO antigens are present on the red blood cells. So red blood cells will have membrane, cell membrane, as we all know that. Cell membranes will have a lot of lipids and proteins. Again, we will talk about that in the lipid lecture later on. But there are glycolipids, and these glycolipids are ABO antigens. Okay, so you must remember this for your MCQs or multiple choice questions in your exams. So ABO antigens means they are glycolipids. We'll talk about that later on little bit in more detail. So these are glycolipids embedded within the plasma membrane of red blood cells. Okay, what about a rhesus antigen or RHD antigen, which is determining us whether we are negative or positive? These are proteins. Okay, ABOs or glycolipids present on the membrane, and RH antigens or proteins that's also present on the membrane. Okay, in addition, the membrane will have a lot of glycoproteins and other things. Okay, as usual, because red blood cells or cell types, and they will have a lot of other different important channels and receptors, and there are molecules for identification of those red cells identity. So they will have channels, for example, allowing any ions to go inside the cell or coming out of the cells. And these molecules present on the membrane are really important to the immune system, because the immune system will recognize any foreign materials and try to produce immunogenicity or immune response against those material. For example, production of antibodies. So our immune system will generally ignore our own molecule, all the self molecules. Whatever the molecules we are carrying in our cell types from the birth will be identified as self molecules, and therefore our immune system will ignore them. However, as I said before, the immune system will recognize the non-self molecules on the surface of cells. For example, COVID-19 spike protein expressed on T cells. It's a non-self proteins because we don't normally carry COVID-19 surface protein or a spike protein in our cell types. So as soon as the infection happens, the T cells will express the spike protein on the surface. Therefore, the immune system will identify that protein as a foreign material and develop immune response. Okay, so any non-self materials will be identified by our immune system, and therefore the immune response will be kick-started. Okay, that's what is happening. So mainly against pathogens or during disease conditions, we might be expressing any proteins. Okay, so here you try to remember that ABO antigens are type of glycolipids present on the membrane of red blood cells, and RH antigens, particularly RHD, or proteins that are also present on the membrane of red blood cells. Okay, so this is how the antigens look like. They are sort of like spikes on the membrane cell or red blood cells membrane. Okay, so if somebody got a blood group, they will have A antigens on their red blood cells. Okay, so we will talk about that in detail later on, but they will have A antigens on their surface. If someone is containing B blood groups, they will have B antigens on their surface. Okay, and if someone is AB blood group, and they will have AB antigens, both A and B antigens on their surface. If someone got O blood groups, they will not have any antigens. Okay, they will have just basal sugars, but not the actual sugar molecule that's determining whether we are A or B or both. Okay, we'll talk about that in the next slide. So these are the composition of the antigens. So A antigens on people who got A blood group, B antigens on people who got B blood groups, and both of these antigens, both A and B antigens in people who got AB blood group. And then in O antigen, there won't be any particular antigen. There won't be any A or B. That's why they are called O antigens. OK, I'll stop this video here and I'll start the next video to talk about particularly about these antigens. Thank you.